Okay. Hi, everybody. Thank you for the invitation. A lot of familiar faces. I'm based at Cornell's Medical School, which is in New York City. And in our group, we use a combination of computational and experimental methods to look at a number of different aspects of cardiac electrophysiology. One of which we've looked at a lot over the years, as many of you know, is Alternans. And so what I'm going to talk to you about today is using control of action potential duration alternans as a means of illuminating the mechanism of alternans. So I'm not going to spend too much time in the introduction. I think most of you are already familiar, and certainly via Wes's very nice talk, it would be a waste of everybody's time to talk in too much detail about what Alternans is. But so this just shows, this is for the first part of the talk, I'm going to show all mostly computational work. This is just a cellular model showing voltage and calcium at faster pacing in Alternans. And over the years, there's been some disagreement and debate in the community about what the underlying mechanism of Alternans is. And there, to try to summarize it, there are essentially two main theories. One is that it's caused by a voltage instability where Alternans arises when, so this is just a schematic of a part of a cardiac myocyte as represented by a computational model with ion channel and components on the membrane and the T-tubule and the sarcoplasmic reticulum, the calcium storage component. And so the voltage instability theory says that Alternans arises when there's insufficient time between each beat for the sarcolemmal ion channels here and here to fully recover. And the calcium instability mechanism hypothesis says that Alternans arise again when there's insufficient time and between each beat, this time for the intercellular calcium handling system here to completely recover. However, because they are bidirectionally coupled, Alternans in one of these systems will cause secondary Alternans in the other. So what we've been interested in doing is trying to, for a given cell, tease out which or both of these mechanisms is active. So these are some experimental data. You'll be seeing more of these types of slides going forward. But this is whole cell calcium Alternans. And what this is so this is just, say, a calcium concentration versus time plot. And these dashed vertical lines indicate the time points at which two images were recorded and displayed here. So this the color is a function of the calcium concentration. Red is a lot of calcium, blue is not very much calcium, and you can see that for this beat you have a lot of calcium everywhere, and for this beat you have not nearly as much calcium at the peak of the action potential everywhere. We're going to be talking a lot about something else called subcellular calcium Alternans, and in this case, again, experimental data. This shows a cell at 2 consecutive beats, but what it shows here is what you can see here is that for the first beat, the nth beat, the calcium at the top half of the cell is at a much higher peak than the calcium at the bottom half of the cell here. So that's this dashed vertical line. But it flips for the next beat. And so what you have is this spatiotemporal pattern where both halves of the cell are in alternation, but they're 180 degrees out of phase with one another. So this has been reported both in isolated cardiac myocytes and intact tissue. And one reason it's interesting is that it may predispose the cell to arrhythmogenic intracellular waves of calcium release. So for the first part of the talk, I'm going to focus on what is the mechanism of subcellular calcium Alternans. And the way that we've probed this is using Alternans control. And I'm going to just blow through this slide quickly because Wes just talked about this method. But essentially, we're going to be, everything that we're trying to do in the lab or computationally are things that we can do in the lab experimentally. And so this, what I'm showing here is computational work, that this is just a single cell model where you have long, short, long. The black would be the next long action potential. But if you use the method similar to what Wes just described, and you basically exploit the inherent action potential duration restitution dynamics of a cell, whereby you shorten the diastolic interval, you shorten the subsequent action potential, you can control and eliminate action potential duration alternans, and this has been done by many groups over a long period of time. And this is just an example from guinea pig myocyte data from our group showing the pacing period, this here called TN, and the action potential duration. So for 200 millisecond pacing period, you have stable alternans, and then when you activate this control algorithm, it eliminates it. One thing to point out here is that although it eliminates it, it never eliminates it entirely, and that's due to sensitivity of the algorithm, biological noise, etc. But I'm going to talk towards the end of the talk about how we are exploiting the fact that there are continuous dynamics here that we can observe. Continuous dynamics in the sense that they keep going, not in the continuous. Yeah, Flavio. Yeah. You certainly can. This is a guinea pig ventricular myocyte, and I think what you find here is that in this case we're able to pace it down pretty effectively so that it continued. 90 to, are you looking here? 90 to 170? This is the APD as a function of beat number. Yeah. Yeah. I don't recall what you mean in the whole ventricular image. Yeah. So that may be the case. So these are all these, like the Rosenbaum work, these are all at room temperature as well. Yeah. Yeah. We see this sort of pattern repeatedly. So I guess the answer is yes, at least compared to. So we it's been quite a while since we looked at tissue level alternans in ventricle. We did a little bit of that a long time ago. So okay. So to try to understand subcellular alternans, we started with a computational study. This up here is I'm calling it a standard model, not the standard model, but a standard model in the sense of you have one bulk calcium component that's modeled by a single set of equations. But that would not enable us to look at spatiotemporal dynamics in calcium, and in fact it's not realistic. Realistically, cells are more like this, where you could hopefully see these kind of gray striations, and those represent the sarcomeres of a cardiac myocyte, each of which has its own calcium sarcoplasmic reticulum system. And so what we did was we enlisted the model that was developed by Johannes and Eigen and used by them and their colleagues in a number of studies for a number of different purposes, including looking at subcellular calcium alternans. So what this model essentially does is it has a series of an independent series of equations for the subcellular calcium system. Now, so there are some important characteristics of this model that are relevant to understanding subcellular alternans. One is that because membrane potential in a cardiac myocyte equilibrates rapidly on the scale of a single cell, on the order of about a millisecond, in this model, it's although we have independent series of equations for each spatial locational calcium, there's still one bulk set of equations for the membrane potential and the transmembrane ionic components. That's consistent with the physiology, so that's okay. Whereas you have the independent calcium, so calcium transients very spatially very slow. So it's something like 100 times slower for calcium diffusion relative to membrane potential diffusion. So you can represent, so the action potential at one end of the cell is essentially the same as the action potential at the other end of the cell on the time scale that we're interested in. But the calcium dynamics diffuse very slowly relatively. Okay, so this is, I'm going to try to talk through the idea behind the computational study. So the effects on local effects on calcium on the membrane potential essentially average, because you have one series of equations and one membrane that has, for the reasons I described on the previous slide. So thus, action potential duration alternans, which is membrane potential alternans, is dependent on the whole cell average calcium alternans. And action potential alternans can therefore only be eliminated if whole cell calcium alternans is eliminated. So let me try to show you that in a couple of the next two slides. So first, I'm going to consider the case where the initial conditions of every sarcomere are identical. So the equations for every sarcomere are going to be identical throughout, but in this case, you can imagine that you can change the initial conditions, which I'm going to do on the next slide. But initially, the initial conditions on every sarcomere are identical, and I'm going to show you now perhaps the most boring movie that you've ever seen in a talk. But what you're going to see here is, so this is essentially a moving graph, and what it's going to show you is on the y-axis, on the y-axis here is calcium alternans, and calcium alternans is quantified as a difference between the peak of the calcium at one beat relative to the previous beat. So initially, we're pacing this cell rapidly, and initially there is calcium alternans. There's significant calcium alternans everywhere in the cell, and they're all identical because the initial conditions are all the same. Then, and this graph here should really sort of be a point where this represents the action potential duration alternans. So that's the difference between the action potential duration at one beat relative to the previous beat. And so that's one number because there's one value for the entire membrane. So initially there's an action potential duration alternans here of about 12 milliseconds, something like that. So then what we're going to do is, so this is pacing rapidly, and then when I activate this, we will trigger this alternans control. And what you're going to see, and what alternans control does, is it doesn't know anything about the calcium. Okay, all it knows about is the membrane. And so it's basically going to use this approach to eliminate the action potential duration alternans, and you're going to see this bar just kind of come down here to 0. And you will see that because you can only eliminate that by eliminating the calcium alternans, you're going to see this all go down to 0 as well, and it's going to just be all synchronized. But there's a reason I'm showing you this, because it sets you up for the next slide. So the algorithm is able to eliminate the action potential duration alternans, and it does that at the same time as it's eliminating the calcium alternans, although it's not trying to eliminate the calcium alternans. Okay, so now let's look at two cases where it's the same dynamical system, but this time we're going to start out with different initial conditions on every sarcomere. And the different initial condition is just simply, we just tweak the initial calcium concentrations. You could do this in a number of different ways. That's one that we chose to do. So when we start out, you're going to see that you could see that the alternans, because of the different initial conditions, are no longer uniform in either of these. So we're going to look at this one first. Again, you have significant action potential duration alternans. It's going to drag that down. Let's see what happens on the left. So what it does is it starts to eliminate the calcium alternans, but brings it all down. But then at some point, because it's non-uniform, part of the calcium alternans crosses over the 0 line, and then the perturbations for the control algorithm are out of phase with what needs to what would be required to eliminate that. So it's unstable. And the stable state that it settles in to eliminate the action potential duration alternans is a subcellular alternans where one end of the cell is out of phase with the other end of the cell. Now, this is another case with different initial conditions. The same premise applies. Watch this is going to cross over here, and then once it crosses over, it essentially is amplified. And now what you have is ideally this should have run a little bit longer, but if it did, what you'd see is a perfectly symmetrical end state where you have the middle of the cell out of phase with the two ends of the cells. And I'm going to show you some experimental data where we see both of these cases. The simulated cell, so in 80% of the trials with different initial conditions, we ended up with this 2-region subcellular alternans. And in 20% of the trials, we ended up with this 3-region. And you never knew ahead of time what it was going to do. Right. So in this, for this length of a cell for this many sarcomeres, we never saw more than 3. But if you went longer, you can get more. It's essentially a Turing instability, as Johannes and Eigen showed very nicely in a paper a few years back. That this work used a lot of their ideas, and then we tweaked them a bit to try to do something that we could do in a real cell. But they demonstrated in a very nice study that it's essentially a Turing instability, a Turing pattern formation. Okay, so I mentioned that we're trying to do things that we can do in the laboratory. So let me tell you a little bit about the experiments that we do for this particular area of research. Yeah. You want to go back?
Yeah, but just that cell. This cell is part of the ring of cells, right?
So this cell is isolated. It's a single cell. But in both the simulations and the experiments that I'll talk about, but obviously in the heart, it's not.
So I'm just thinking about this because what's that V that you're changing? It's your stimulation, right?
Mm-hmm. Changing so we're stimulating periodically that cell fast. You have alternans, and then what you do is no, your stimulation is periodic.
Okay, so why does the cell show alternans?
It shows alternans in membrane voltage because let's go back. Let's see. Bear with me for just a second. I can show you this, I think. Let's see. Oh, I did that wrong. I'm not as... So it's essentially it's a dynamical system that has a period doubling bifurcation. So you have this shows action potential duration of a model. The action potential duration is going to shorten as a function of the cycle length. Okay, is everybody okay with this, or okay skipping this? All right. You could see the energy well kind of analogy there.
So the model you are using, is it with the stochastic counting?
No. So it's deterministic with the exception of the initial conditions.
So without the stochastic noise.
Right. Of course, if you had that, you should get the same thing, but you don't need that if you throw some noise on the initial conditions. So this shows a heart from a. Let me see here. Is it a battery? No. It's okay. Let me just use a pen or something. Oh. It's too high tech. This shows a heart from a guinea pig. And so the guinea pig is as most or all of you know, is a very good small animal model for human cardiac electrophysiology. And so you isolate the guinea pig heart, and this just shows an apparatus where you Langendorf perfuse it, and you exploit the inherent cardiac coronary vasculature to pump through collagenase, protease to digest the cell, and you get a series of steps over a period of several hours where you can then isolate individual cardiac myocytes. And then once you do that, you take the individual cardiac myocytes to your rig, and we patch clamp the cell and calcium image the cell. And we do these experiments at room temperature because guinea pig cells don't show much alternans at physiological temperature. But if you're thinking of it from the perspective of a dynamical system, I think that's okay. So what this is going to show, this is a movie that's going to show you alternans in a cell, calcium alternans in a cell. And these two regions are identified after the experiment. So you don't know ahead of time where the subcellular alternans is going to occur. When I start the movie, you will actually see something in these boxes. White is going to be calcium fluorescence, and black is no calcium. And so initially, so this is at static pacing, rapid static pacing, and this is the calcium alternans, large, small, large, small, large, small, and the whole cell, both ends of the cell are in sync with the whole cell. Things are scaled here, or normalized. Now, when we run it, what you will see, so you see everything synchronized, and then now we turn on alternans control. What you'll see is alternans control is eliminating action potential duration alternans, which we don't have a visual of here. But as I mentioned, in so doing, it can only eliminate action potential duration alternans if it eliminates whole cell calcium alternans. And you see that as it's starting to eliminate the whole cell calcium alternans, and it's accomplishing that by throwing the two ends of the cell out of phase with one another. So you have a very clear subcellular alternans where the two ends are out of phase with one another. It can't. It can try, but that state is it's an unstable state. So when I showed you that very first computational simulation with no noise, it's able to achieve that with no noise. But if there's any little bit of noise, it can't find that state. So I'm going to show you another example, and this is a 3-region subcellular alternans. And when this movie is going to start right at the end of alternans control, and so you will see subcellular alternans in 3 regions, the two ends in phase, out of phase with the middle, and you can actually already see that here. You see there's maybe very small, or not maybe, but there is very small whole cell calcium alternans, but you see that the red and the green are in phase, but out of phase with the blue. So alternans control had been run here, and then the purpose of this movie is twofold. One is to show you a 3-region subcellular alternans, and the second is to demonstrate from a dynamical systems perspective that the algorithm is not fundamentally changing the system. It returns back to its initial state. And so initially you can see the 3-region subcellular alternans. And because now we're simply just static pacing, that's no longer a stable state, and it's going to settle back into a whole cell alternans, which is its stable state at this pacing rate. So we did this in 16 different cells, and on those 16 different cells, 51 trials. Of those 51 trials, the alternans control was successful in that it was able to suppress the APD alternans in 45. Of those 45, 42 of them showed subcellular alternans, and this was done at varying cycle lengths. None of the trials in which control failed showed subcellular alternans. 6 of the 16 cells tested had at least one trial with more than one period 1 calcium node, and I'm going to talk about that more on the next slide. And subcellular alternans always coincided with the elimination of whole cell calcium alternans, and I would contend that it caused it. So maybe you're forming the opinion that the subcellular calcium alternans is a dynamical mechanism as due to some fixed anatomical heterogeneity. And we believe that to be the case, at least in these particular cells, in this particular experimental approach. And here's evidence as to why. So this shows one cell in two different trials. So you do a cell, you wait, and you do the same cell again. And it shows that in one of the trials you had the 3 region, and one of the trials you had 2 regions. So 5 of 16 cells showed the ability to have both of these behaviors. So if there's some anatomical structure that's causing the this is inconsistent with some fixed anatomical structure causing the subcellular alternans. And when we talk to this to biologists, they usually start by thinking it's anatomical, but I think they get convinced by the time we get here. I'm going to skip the other parts here so I can get to one more part of the talk without taking too much of your time. The next 3 slides are the most recent work that we've done on this particular topic, and it's what informed the title of the talk, which is using alternans control to illuminate the underlying mechanism of whole cell alternans itself. So theoretical studies have shown that the presence of subcellular alternans indicates that calcium instability contributes to whole cell alternans. So there's a number of studies. Johannes and Eigen and some work from our group and others have showed that the subcellular alternans, for example, as I showed on the previous slide, cannot happen if there's not an inherent instability in the calcium system. So I'm not going to show you all of that work, but that's been very well established. But what really has not been as well established in its experimentally, it's difficult about whether or not there is a voltage contribution. So a long time ago, it was the idea that voltage was causing alternans, and then really the predominant theory now is that it's calcium is causing alternans. And I'm not going to show you anything to suggest that calcium is not causing alternans, or at least the main component. But I am going to show you some evidence, I think, that demonstrate that voltage is playing a role as well. So this is a modeling part of a recent study. So it's a busy slide. In fact, all 3 of the slides are kind of busy. I hope I can get through them without too much time. So this is using the same cellular model that I talked about before. And this shows actual duration as a function of cycle length, and the black is if you, the gray is if you stimulate this without any control, and you get this period doubling bifurcation. You have actual potential duration alternans. The black is if you apply alternans control, and what you do here is you basically stimulate at this rate, and you have activated the alternans control algorithm, and you slowly ramp down the basic cycle length without ever turning off the alternans control. In the chaos control community 10, 15 years ago, this was referred to as tracking, where you are essentially tracking the unstable period 1 fixed point. So if you do that, then this is a metric measuring subcellular alternans at each one of these basic cycle lengths. So out here, before you have alternans, even you don't have any subcellular alternans. And then you have subcellular alternans, and then in this particular model, and I'll talk about which model this is on the next slide, it goes away. This gray region here, which I hope you can see, it's a box, a gray box that sort of goes from 200 up to about here, is the region where the actual potential duration restitution slope is greater than 1. And so how did we get that? So if you jump over here, this is the actual potential duration as a function of diastolic interval. If you remember back when I showed the very first control slide where I said it never really goes away entirely, there's still some dynamics there. If you take every one of those points, during control and you can get an APD versus preceding diastolic interval, and the gray points here are the data, the computational data, and the black is a fit to that data, and you can take the slope of that. So you can get a restitution curve you can get a restitution curve in a region where it's very difficult to get it without control. And the slope of that is here. The region where the slope is greater than 1 is here. And the reason that's important is this also a very busy slide. And so there are 4 different models here, these vertical columns here. And these are 4 different variants of the model that Johannes and Alain developed. So that model's, for a number of reasons, is very nice. But one of them, for this purpose, is that you could tweak some parameters in the model such that the alternans, in this case, is caused only by calcium. In this case, is caused only by voltage. And these are 2 cases where they both have a contribution. And so then what we did was we applied Eigen mode analysis. So thanks to Niels, developed this technique as applied to alternans, and what Eigen mode analysis demonstrates is it's basically a theoretical measurement of the instability of the system. And we did that in three different modes. One was where the system was paced. Those are the circles here, and that's you get a measure of the whole cell instability. One is where you do an action potential clamp, and those are the black bar dots here. And that if you do an action potential clamp, so basically you fix the action potential to its appropriate morphology, and then any instability cannot be due to the voltage. It's got to be due to the calcium. So this is a measure of the calcium instability. And if you calcium clamp, similar to action potential clamp, you can't do calcium clamp experimentally, but you can do it in a model. You clamp the calcium to its morphology, and you can get a measure of the voltage instability. And so what this shows is that the cell, as one would expect, anything less than -1 is unstable. So the cell is unstable for everywhere where it bifurcated, as you would think. And then it shows that initially it's a voltage instability, and then voltage becomes stable, and it becomes a calcium instability in this particular. No, I said that wrong. I'm sorry. Initially, it's a calcium instability, which is required to have subcellular calcium alternans, and then eventually you actually get voltage instability. So they have different kinetics as a function of rate. So you could do that for all of these. I mentioned that. So this dashed vertical line is the Eigen mode calculated calcium instability onset. So the theory predicts that the calcium instability is going to onset at the exact same point, in fact, where the subcellular alternans start. So that's good. That's what I just mentioned. What we also find is that you get a close but inexact, and I'll talk about it in a second, match between the Eigen mode voltage instability calculation and the slope of the restitution being greater than -1. So why do we care about this? The reason we care about this was, I mentioned a second ago, experimentally we can't measure these Eigen modes because we can't do all these we can't do the calcium clamp, but we can measure this restitution slope. So the purpose of this was to demonstrate that this method, using the restitution slope and the subcellular alternans, is consistent with the theory, and therefore it's an experimentally viable method. If we can do the experiments, experimentally viable method to measure these. Okay. So this is basically what I just said. So APD alternans control and subcellular calcium alternans detection provides us with an experimentally feasible method of illuminating the presence of both the calcium and the voltage instabilities. This is the last data slide. These are the results of 5 experiments on 5 guinea pig cells. So it's experimentally feasible, but it's experimentally very difficult. We seem to have a knack of developing methods that are just really, really hard to actually do in the laboratory. But fortunately, the postdoc who did this work, Willemijn, stuck with it for a long time and it was able to get these data. So what this shows is the subcellular alternans, and again, and it shows the region where the restitution slope is the magnitude of the restitution slope is greater than 1. And so so for all, you have subcellular alternans in all 5 of them when you do control. So calcium plays a role in 5 out of the 5 cells. So you had all these cells had alternans, and you control it, and you see subcellular alternans. So we know that calcium plays a role. Voltage plays a role in 4 out of 5 of the cells. So in all the regions where you have alternans, the slope of the restitution curve derived from the control perturbation data is greater than 1. In cell all but cell number 1, so the slope of the restitution doesn't get greater than 1 until after you're no longer basically able to control. So this was entirely calcium mediated. The subcellular alternans actually almost went away towards the end, but not quite. Cell number 2 is a good example of how voltage and calcium roles can vary as a function of rate. So initially you have the alternans is entirely calcium, but then as you get to faster basic cycle lengths, both are making a contribution. So in conclusion, in model cells and isolated guinea pigs myocytes, alternans control induced subcellular alternans robustly. Those are the experiments from most of the talk. The results strongly suggest the mechanism of subcellular alternans, at least in this system, is dynamical rather than anatomical. And then what I just showed you, subcellular alternans coupled with action potential duration restitution measured during control can be used to determine if calcium and or voltage underlie alternans in myocytes experimentally. And these are the published references for the various aspects of the work that I talked about, in case you're interested in seeing more details. And these are the people that did all the work. Most of you probably know Trina. She's here, and she's been involved in all these studies in a very important and integral way. Steve is a very talented former MD PhD student in the lab who did the experiments for the first part, the subcellular alternans experiments for the first part. Willemijn's a very talented former postdoc who did those experiments and the computational theory stuff of the last part of the talk. Francis is a former lab tech, now graduate student, who's been involved in all these studies. And 2 colleagues who were very helpful, Jeff Abbott, and many of you may know Gil Bubb, who was very helpful to us as we were setting up our calcium imaging stuff. So thank you for your attention. I'd be happy to answer any questions.